Okay, so it should be recording now, so this is good. So let's start off with the main concept. So why are we learning the Central Dogma? Now, for those of you who remember the lecture, the idea behind the Central Dogma is that this is this kind of universal way of understanding how life works. And really, we try and teach it backwards because it seems to make sense to most. And so the story starts with this idea of proteins. So I'm going to start it backwards. So I'll write it up. And you'll also notice that I'll try and color code them just so that they can make a little bit more sense. Now, proteins pretty much are all functional units, meaning these are the things inside every living thing that we know of that performs a certain job. They do a specific function. They do stuff. Now, what type of stuff do they do? Pretty much the answer should always be everything. Proteins are the ones that do everything in living things, with a very minor adjustment of about maybe 2-3% of the jobs that are done inside a living thing being done by RNA. Everything else is done by proteins. So the question then is, how do we get those proteins? Since they're important to us to do everything inside of us, they're pretty much doing transportation, conversions, all the enzymatic reactions. They help us produce energy. They help us in our immune system. They do everything, basically. So how do we get them? How do we make proteins? And so ultimately, our proteins have to be basically encoded somewhere, programmed somewhere, if you will. So the code or the instructions to build proteins comes in the form of DNA. So this is why we call it the blueprint. And what you'll also notice is that usually I end up coloring that one also in a little bit of blue. Kind of goes with it, right? So the idea is proteins are the functional piece, and then DNA is the code to make those things. So in the lectures, I usually kind of explain about, think about it as anytime you buy furniture from Walmart, IKEA, or something like that, and everything you're about to build has to come with a series of instructions, and then you kind of read those instructions, and then ideally you build it with some sort of materials. And so the same thing applies here. DNA happens to be those instructions. And I'm going to emphasize it right now because that's kind of like the killer line at the end of the lecture too, is that ultimately if you don't know how to read those instructions, then what you end up building ends up being poorly built. So that applies to us as well. So you need a really good set of instructions that you can follow so you can build really good proteins. That's the idea. Now, the problem with this is that you can't build proteins directly from DNA. Right? You need something to help you to do so. And the idea here is kind of, if I relate it to, for those of you who've ever seen actual blueprints, you know that most of us can't really read a blueprint. We usually need some sort of expert, somebody to kind of tell us how to read those things so that we can actually put it together. And so this comes into place of things like architects and contractors that know how to read a blueprint and do that. So for us, the one that helps us decode the DNA, so decode those instructions, those blueprints, is RNA. Now, there are a couple of different variations of RNA, so we'll cover that in a little bit, but the main one is known as messenger RNA. And it's commonly abbreviated as mRNA. And so for the purpose of us, I usually kind of highlight that in red. So it kind of makes my little job a little bit easier. So I'm going to do the same thing with this guy too. Okay, so here I kind of covered all three main components. We have the functional pieces of life. So you, bacteria, viruses, everything needs proteins to do stuff, right? And then the codes the blueprints, the instructions that they come with, that's going to be our DNA. And then the in-between, the one that allows us to convert DNA ultimately into those proteins. Now, I'll let you in on a quick little secret, and I'll add this in a little animation over here. Let me get that. The process of turning those genes into actual proteins is known as expression. And more often than not, it's called protein expression. So it's the same thing. So whenever any scientist, any expert, or even in your textbook is talking about, hey, how do we build proteins? We use the term expression of proteins, meaning that we're taking our DNA and turning it into protein. So that's called protein expression. Okay. So let's take that idea first of how do we get our instructions to make those proteins. And so for that, we kind of have to start clarifying what each is. Now, again, this takes much longer usually in the regular lecture, but let me give you like the little pieces that should be important for you in the exam. So let me get to the next slide. And let's start off with the instructions. So I'll start with the blueprint. The blueprint, in this case, happens to be a collection of instructions, not just one, right? A series of instructions all kind of packed together. And so we call that DNA. And so the easiest analogy or even metaphor that comes to play when I describe this is that of a library. So a library happens to be this collection of knowledge, right, that you're supposed to access. Well, the same thing applies to DNA. DNA is not just one instruction for one thing. DNA happens to be an instruction, a collection of instructions. And so that collection is usually referred to as the genome. So whenever you hear somebody talking about a human genome or a bacterial genome, that's what they're really talking about, a collection of those instructions, right? So the genome, as its name implies, is a collection of genes, meaning individual instructions to build proteins. So what are we talking about here? Well, ideally is that if you went to the library, let's just say, you would be going there for typically some sort of purpose to look for some set of information, right? The idea is that you go in there to, say, look up science fiction or look up how to build houses or look up, how to fix a car. There's always going to be a book or books for that particular set of information you're looking for. But ultimately, all of that information is contained within that library. So if I wanted to build a particular protein, if I wanted to do a particular job per se, I'd go to my library and go get that information. So if I want to build, for example, insulin, which happens to be a protein, also happens to be a hormone, I would go to my library and say, hey, where do I make, or how do I make insulin? And in my collection of books, I'd read up the book on making insulin, and bam, I now know how to do that. So DNA is often kind of mistaken as this just kind of little bundle of stuff. And in reality, it's a really, really cool and very, very efficient way of storing all that information. So if I can give you some sort of mini analogy, let's try this again, is that think about your library as your genome. Think about each, in this case, each book as one gene. And think about the knowledge that you're getting from each book as one protein. So if I want to learn how to make a protein, a specific one, I go look for a book inside my library. That's the idea. So what we're going to show you in this case is that your DNA, the part that I'm going to be highlighting in blue a lot, happens to be that collection of all these sets of instructions. Okay, all of the proteins that you can make are all stored within that. So as a silly little example, let's just put over here. Ours is in microbiology, but most of the examples can be compared to humans. So one of the most common examples is that the human genome, meaning the human library of these things, contains roughly about 25,000 genes. So you want to think about it as if our library contains 25,000 books, basically. And each one of those books you can read and you can get a specific set of knowledge. So for example, as a sub-example, in this case, one of those genes could be the gene for insulin. In other words, one of those books within our library tells us how to build insulin. So what needs to happen is we go look for that gene, learn how to make that, and then make it. That's the idea. You go look for the instructions and you learn how to build it. So in this case, since the gene is called the insulin gene, we learn how to read it. So let's call it a book of instructions. And then build insulin based on those instructions. And so insulin for us happens to be proteins. A protein, I should say, actually. A protein called insulin. Now, we don't only have one. Like I said, in humans, we have about 25,000 of those, right? But every organism is different. So, for example, bacteria range between just a few hundred to a couple of thousand only. Plants can have up to 50,000 of these guys. And so it really kind of varies from species to species, from animal to animal, from even related organisms. There can be some variation associated with them. So the idea here is, again, the collection of those instructions, our blueprint, we call that DNA. Again, it's our collection of instructions. Now, if you want to see a different analogy that I usually provide, is think about if you ever wanted to build a house, literally like using a blueprint. The blueprint includes the instructions for everything, not just, for example, a bathroom. The instructions in the blueprint tell you how to build bedrooms, to build bathrooms, piping, wiring, where do doors go, windows go, everything is collected within that set of instructions. And then as you read each piece, you get each one thing. So the same thing applies here. We need to understand how to read that blueprint. Now, let's take it to the next step. Let me advance it, and we'll come back and forth between these guys. Is the other piece is that not everybody can read a blueprint. I can spell blueprint, right? You typically need some sort of expert to decode that. Right? Most people, most of us don't know how to read a blueprint. So we need to find somebody that can do it for us. So again, somebody like an architect, somebody like a contractor, somebody that knows that particular field. In our world, that expert is called RNA. And to be a little bit more specific, it's called messenger RNA or mRNA. That's the guy that knows how to read that guy. Okay? Now, let me bring this down a little bit. Our messenger RNA basically uses the DNA template, the DNA strand as a template. So let me color code this guy. So our RNA, roughly, I always keep it in red, and our DNA, I always keep it in blue most of the time. Right? So what this expert is going to do, this mRNA is going to figure out what to build out of that DNA. That's what it's going to try and do. Now, once it decodes it, in this case, then we'll be able to build what we want from it. So the first thing we need to do is understand what that DNA means. Okay? So this is where mRNA comes into play. This is where the RNA comes and figures out what that library means. Think about it this way. Most of the time, if you were to go into a library and pick up a book, it is quite possible that some of those books you don't know how to read. Not necessarily the words, but in terms of the content. Sometimes you require a little bit more expertise on how to read certain technical books, or even science books, for example. So you need somebody to help you understand that. That's what RNA does. It helps us understand what the DNA really means. Since DNA is long and complex and incredibly combined in terms of its alternation of the 4 bases, you get tons and tons of options. So you need a little bit of help. That's where RNA comes in. That's our expert that helps us more or less decode what the DNA wants to do. So that's where that guy comes into play, right? So we'll keep on going with this idea. I'm going to give you a new slide really quick. And so ultimately, once we read the blueprint, now we want to build our product. In other words, we want to make protein. And again, how are we making that protein? By going to the library, by looking up the instructions on how to build it, and then using some sort of expert that tells us how to build it. That's our RNA. And then we ultimately build that protein. So that's where the kind of 3 steps of the central dogma comes into play. So now, then we need to use our decoder, our interpreter, if you will, our mRNA, so we can build protein. And so that process of building it requires materials to build it, just like anything else you want it to build, right? You need some materials to do so. And those materials to build protein are called amino acids. Now, let me color code these just to kind of continue with our theme here. Getting these nice and colorful. And so, as I'm pretty sure most of you are familiar with it, you've heard, proteins are made of amino acids. That's what we learned in even topic B. And now we're just kind of further emphasizing it. So to kind of hone in on a little bit of these topics will haunt you from the very, very beginning. Reminders of topic B. Remember, we had these things called monomers, and we had these things called polymers, right? And so one version of these were your amino acids that when you assemble them through dehydration synthesis, you obtain proteins. So we take the subunits and build them into the final unit product. So think about this again in this little analogy, if you will. Whoops, I deleted that guy. So our analogy once again is that you would have your amino acids, which I'm going to abbreviate from this point on as little AAs, right? Could be things like your wood and nails or something like that. And then ultimately your proteins would be something like a chair, right? So you have the materials to build them and then the final product that does a job. In this case, allows you to sit or something like that, right? The thing is though, again, how do you build that chair? It's fine and easy to go buy the stuff, but to build it takes a little bit more. And so to actually put those nails and wood together to actually get something productive, you need instructions. And remember, as we just kept on telling you, those instructions come from your library. of instructions. In other words, your way to build those materials, in this case our amino acids, come from our genome collection of genes. So what are we trying to do here? We're trying to kind of show you that there's an actual flow of information. In other words, let's go back really quick. You need to start with a series of instructions that tell us how to build things. That's our library of genes, or what we call DNA, or the genome. And they tell us how to build these products, like a chair, like I was mentioning earlier, or an actual protein like insulin. And so we have a collection of these instructions that is our ultimate blueprint. And for us, then we need somebody to kind of help us understand what those blueprints are. So somebody to help us understand those sets of instructions. That's where RNA comes into play, right? And then ultimately, once we understand it, then we can use amino acids, so the components, the materials, to actually build what we want to build. In this case, we want to build proteins. That's the idea. So let's put that kind of all together into a little bit more of a useful visual set of instructions, shall we? So let's go back to this. Let me add one more slide here. Let's go back to, again, the central dogma. And I'm going to take it a little bit longer just so I can kind of color code it a little bit better. But let's kind of start off with our idea. So first, we need a set of instructions. That set of instructions is what you call DNA. Reminding you again that it's a collection of genes. Not just one, it's hundreds, if not thousands, if not tens of thousands of these little instructions. So again, like books. Now, we also need an interpreter that helps us understand that collection of genes in books. So that's your RNA. So this is the decoder of those individual genes. And then once they get decoded, then now we have a way to actually build our final product, which is the stuff we're going to use. So these are the functional units, the actual particles, the molecules that perform the jobs inside a living thing. So functional units of life. And those are called proteins. Okay. So as I coded it earlier, our collection is in blue, our interpreter is in red, and our final product is in green. All right. And so let me kind of switch the layout briefly over here. So let's try this again. So our library of this stuff, our blueprints will be our library, right? Which is a collection of books, right? Then we need our interpreter. That helps us understand those books. And then we have the actual product. What we wanted to build for a specific purpose. So that's kind of bringing it kind of side by side. Okay, so now that we have this idea of what the central dogma really is this kind of flow of information from the set of instructions to a final product, let's kind of illustrate that by going through kind of an example, and then ultimately more of a detailed discussion of what each step entails. So let's go with the overall format, right? So for this, I'm going to be drawing a little bit more. overall. So you're going to see me kind of switching around with some colors, so be patient with me over here. So here we go. Let's just say, oops, that's not what I want to do. Hang in there. That what I want to build, I'm going to put them side by side. And so you're going to have to forgive my writing skills over here. I'm kind of going to divide my screen into two pieces. So let's do this. Okay. And so on the left side, we'll talk about biology. And on the right side, we'll talk about life, even though they're related, right? So let's just say, let's put these kind of examples. And since I'm doing this already, might as well kind of go with the flow over here. Let's just say that in real life, I want to build a window. Okay, so I want to build a little window. I'm just going to do this. So don't judge my artistry. Okay, that's a window, people. Go with it. And let's just say that in biology, like I said before, I want to build this awesome little protein that does this cool little job called hemoglobin. So in real life, we want to build a window. And in biological life, we want to build hemoglobin. So again, these are things that ultimately perform a function. They do a job. One of them keeps us alive. The other one kind of keeps the bad guys from coming into our house, kind of thing, right? So now, I assume that unless you're in the business, most of us don't know how to build either a window or hemoglobin. So what do we need? We need instructions. So the very first step in here, let's label these. These are going to be our products. So I'm going to write it off to the side. Our final functional products, the ones that do something for us. And up here, I'm going to put in the instructions. So I need the instructions on how to build hemoglobin, and I need the instructions on how to build a window. So I would basically, in biology, go reach out my DNA. So I'm going to draw it out over here. It's a cool little double strand that you all remember, right? Not that my artistry is going very well right now. Don't judge me, right? And in real life, now we have the internet, but back in the good old days, like my age, in this case, you'd go to this cool little building, you know, called the library. That's a building, don't judge me. Okay? So that's where you go get them. Now the thing is, you don't go and read all the books. You don't need that. You just need to read one. And the same thing applies to our DNA. You don't need to read all the DNA to build hemoglobin. You just need one little piece. So for this, you want to check out one book. Okay, one little book. I'm going to attempt to draw a book. Okay. This is doing it with a little tiny pen over here. Right? You'd go check out one book on how to build windows. So same thing here in DNA. You don't want to take out all thousands of these guys. You just want one gene, one little piece of it, the gene to build hemoglobin. Now again, most of us are not experts, so we don't know what we're doing there. So we need somebody to help us out, you know, somebody that can help us kind of decode what's in there. Somebody like an expert, somebody like an interpreter, somebody that is specialized in that field. And so for that, we need a little bit of help. So I'm going to put this here. And let me color code this again, sorry. We need a helper. And so what will happen is that one gene, not the whole collection, just the one little piece, will be decoded. That was a D, right? And so that means somebody will kind of help us interpret that book, you know, like a teacher or something like that. Somebody helps us understand what it is. And so in the biological life scale, that gets turned into RNA, a little chunk of RNA. And in the life scale, somebody more or less teaches you a cool little lesson, you know, a little PowerPoint on how to build windows, right? they kind of illustrate that by doing a little tutorial. So now that you kind of get an idea of what to do, then you can actually take that final interpreted message and then ultimately turn it into the final product. Same idea in real life. Now that you've kind of seen how to build a window, then you can go ahead and do it. So what do we need now that we have the pattern? In order to build hemoglobin, like I said earlier, you need materials. And those materials are called amino acids, which happen to be about 20 of them that we know that are naturally occurring. And for us to build windows, I assume we need some wood and some glass and some other stuff that I don't know, apparently. I've never built a window, people. That's not true. I have installed one, but not actually built one. So anyway, so we have what we need. Now the question is, what is going to help us to build that stuff, right? And so the process of actually building that protein, so using that RNA to build that protein, we call that translation. For us in the real world, it's literally just building it. Okay? That's what translation means. It's building that little protein, right? Now, the process of decoding the instructions, the process of interpreting, we give it a fancy name. We call that transcription. So for us, that is that interpretation for us. That's somebody helping us understand that book, right? So now, kind of putting together everything that we just saw so far is that in the Central Dogma of biology, we have a collection of instructions called DNA. That's our genome. We'll pluck out one small set of instructions, not the whole thing. We'll decode it by turning it into RNA through a process called transcription. And then we'll take that decoded material and then translate it, basically build it into that final product that helps us, in this case, carry oxygen and iron to keep us alive, right? By using some cool little materials called amino acids. Or in the same way, we'd go to a library to pick up a specific book of instructions that we need to learn how to read that ultimately will allow us to build a final product like a window. That's the concept. Now, what's going to happen, though, is I'm actually going to duplicate this slide. I'm going to kind of shrink things around just so you know. That way you can see the scale. So hang in there. The screen might disappear for half a second. Okay. So I'm going to duplicate this slide. So let me duplicate the slide. There it is. And so if we go to the next slide, hang in there. It's going to look a little bit different for you guys. So let me pause this guy for half a second. Because I need to keep those cool little annotations. Now, I duplicate that slide. You guys are not seeing this. This is just kind of happening in the background. And I'm actually going to shrink it. You're going to see the same thing, just in a much smaller scale. So let's try this again. Here we go. And sharing with you guys. Same screen that you saw before. Now, what I'm going to do is basically, like I said, shrink it. So if we go to the next slide, if this thing lets me, of course. There it is. I just made what I just showed you in a tiny little piece. Now, what I want you to understand about this scale is actually quite important. This image, let me shrink it a little bit more, make it a little bit tinier, is only one specific case. This is not the whole perspective. There's more to that. We're not made up of just one protein. Humans, for example, are made of about 150,000 of them. So envision this whole process now taking place 150,000 different times. To ultimately build an organism. Now, ultimately, the same idea applies to your house, right? A window is not going to be enough to build a house. You'd have to continually check out all sorts of books until you get all the materials necessary to build a full-blown house. So you need instructions on a door, instructions on a toilet, instructions on a kitchen, instructions on every little piece. In the same way that you need instructions to build an entire organism. One little piece of hemoglobin doesn't build a human. You need all sorts of cells, all sorts of proteins to build that human. So the DNA happens to be this large collection of all the instructions so you can build this entire organism. And again, in the same way, your library happens to be this entire collection on how you can build an entire house too. So now let's take those steps. Let's go one more slide. I'm gonna go over here so I can write on this. I think I might have lost one slide. Hang on. Hang on, people. It's not listening to me. There we go. Now it's listening. First concept that we want to take here is this basic idea of the information. So the instructions, in other words, the DNA, okay, it's not enough to just have it in one place. Just like most things, you need to share information. Just like we were doing in the classroom. It's not enough that I know this stuff. It's important that you know that stuff, right? And so for me to pass on that information to you, I need to literally make copies of this, right? So in our little world of sharing information, we call this copying the DNA. And you know this in a fancy term called replication, right? So for me to be able to build more libraries and more resources of information, there needs to be more copies of those exact same books. There needs to be more copies of that entire collection of information on how to build houses, if you will. Okay, so that process of making copies of it is called replication. So the first process, in this case, it's a very simple idea. You're going to take one double strand of DNA, so we abbreviate double strand in lowercase, in case you're wondering, and literally convert it into 2 copies of double-stranded DNA. And I put the letters backwards. Hang on. There we go. And so how it's done is basically by splitting the DNA open, a process that, for those of you who recall the lectures, it's called melting. And so when you melt the DNA, you can use each single strand of DNA as a template to build a new one. So what does that look like? Well, then allow me to kind of play around with the slides again one more time. Just need to move some stuff around. There we go. And so over here on the right-hand side, I'll try and do the same thing. So let us envision some very basic shapes. And so I'm going to use this by using arrows and doodles just for the sake of making it a little bit easier. So allow me to kind of change colors around and everything else, right? So we talked that DNA happens to be blue for our labeling purposes. So I'm going to make that like that, okay? So let's pretend that that's our little strand of DNA, okay? Now, remember that DNA is double-stranded, for those of you who remember your lectures, right? And it also happens to be anti-parallel. So there's 2 strands of this guy. One going in an opposite direction. So this is our double strand of DNA. Now, it's not really intertwined like it normally would be, like a cool little helix. So for the sake of right now, I'm just kind of straightening them out. Now, what will normally happen is that we'll take these 2 strands of DNA, and the next step is we'll melt them, basically. In other words, we take both strands and separate them out. me add some more arrows and stuff like that we can continue along. Okay. And so this process of separating out the two strands from each other is called melting, or DNA melting is the term for that. Now we're going to use each one of these now as templates to build a brand new strand. So for the top one, we'd read what is there and ultimately create a brand new version of it, which is virtually identical to the opposite side. So I'm going to label this as a slightly darker version of that. So that one gets its new copy. And the one on the bottom also gets its brand new copy in the same way. And so what ends up happening is once we split up that DNA, each of the old strands served as a template for building a new strand. And then what happens is that each one of those ends up going inside a cell. So let me kind of take this guy, and let's just pretend that these two will go into one cell, and these two will now go into another cell. Like that. So let's pretend we're going to make up some cool little cells really quick over here. Each one goes to a different cell, whereas originally these guys came from one cell. And so what we're taking in the process of replication is one double strand of DNA, splitting it open, and using each one of them as a template to construct a brand new one. And so ultimately what happens is those two strands of DNA, one old one and one new one, let me make a copy really quick. There it goes. End up going into the one new cell. I'm going to toss that guy in there. Not the best drawing, but you get the picture. And then the other double strand ends up going to the other new cell. Right? And so what this is, this terminology that we use, which is what the question was asked earlier, is called semi-conservative replication. Conservative would usually say that the two original strands that came from the first cell, these guys, right, the ones that are still kind of the light blue that came from the original cell, would then go back to its original cell. And they don't. Rather, they kind of end up being separated or split to be shared between the other two cells. So it's not conservative replication, indicating that we're keeping the original strands together. It's semi-conservative, meaning that we're only keeping half of it as the originals and half of it as the new ones. So that's where that kind of story splits off. So we're taking the information that is coming from one cell, one little double strand of DNA, separating it out, replicating it by creating two brand new strands for them, and each one of those new strands, together with an old strand, will build a brand new cell, effectively taking one cell and converting it into two. And if you want to add a little bit of flavor to what you just learned, this is slightly more detail of what you learned when you learn about mitosis, for example, taking one cell and splitting it into two identical cells. So before that happens, for those of you who remember, your S phase of mitosis, this is the synthesis stage. This is when you're building the brand new set of DNA, splitting it off into two brand new cells. That's that little story right there. Okay, so by covering semi-conservative replication, we are discussing this idea that we're going to take one mother cell, one original cell with its own single copy of DNA, and then as it replicates, as it starts dividing, the two DNA strands that it contains get separated and then get used as templates to create brand new strands that you're seeing here in our darker blue color. And each of these combinations of old and new strands are what ultimately lead to brand new cells. And so this whole process is what we call replication, and more specifically, in this case, semi-conservative replication as an example. Okay, so let's add that there. Let me add it to my little story over here. So the same process that we just explained, as one DNA strand being turned into two copies of DNA, this is what we call semi-conservative replication. Now, for those of you who want to recall this, this is our very first step in the central dogma. Taking DNA and making more DNA out of it. Okay? So let's proceed. Let's take it again and now establish part 2 of that story. Right? So if we're going to take that, I'm going to create a new slide. We're going to take now those instructions and we're going to do the decoding. Now I need to move the slide, so hang in there. It's not listening to me, so let's. There we go. So now that we know how to make more copies of DNA, now let's make use of it. So using the DNA. So to use it, remember, it's fairly complex, so it's not easy to understand. So we need to kind of interpret it. We need to decode it. So to decode, we need an interpreter. And that interpreter is called RNA. So what's going to happen here? We're going to take one strand of the double-stranded DNA, all right? And this one's usually referred to as the template strand. This will make a little more sense a little bit later too. So take that one strand and then use it, again, as its name implies, as a template to build a new one. Now, mind you, we're going to build, in this case, that new strand using that template, but only a piece. And this is the important part that you need to take into account. We're not converting every single piece of DNA into RNA. We're only building one section of the DNA. Now, what are those sections called? You already know this name. These are called genes. We're only going to take the one piece that we need to build out of. So again, we don't go in immediately into the library and build everything that's in there. We're just going to go for the piece that gives us a chair or a window or something like that. So we're only selecting the target that we need. So in this case, we're going to look for one gene. Let's just say hemoglobin again. We'll go with that. So we'll build one section of the DNA, in this case, the little piece of DNA called a gene, and turn it into a piece of RNA. And so this is called transcription. Now, I'm going to take the images that we did from the previous slide just to kind of help you out with this. So hang in there. I'm going to kind of copy and paste stuff. So here's our DNA again inside a little cell. But in this case, now again, we really don't need both strands. We only need one of them. And so what we're going to take, allow me to add a little bit of labels over here. So we're going to take this strand called the template strand. And I'm going to take it down here too so I can make a little bit more sense. I'm only going to bring in just one of them. Let me see if we can drag this. Here we go. All right. And more than anything else, we're only going to take a segment of it. We're not taking all 25,000 genes put together. That would be just a waste. So we're only going to select a segment of it. So let me kind of put a little square around this area. I'm going to select a little piece of it. Now let me kind of change this around so it makes a little bit more sense. All right, so I'm only going to select a little piece of that. And so that little piece, let me make sure I can edit the text. That way I can continue to add into it. It's not letting me write into it. Hang on, people. I don't know why it's not letting me. It doesn't like me right now. There it is. We're going to call that one gene. And I'm literally going to write one gene so that we can prevent confusion. So we're just going to select one, not the entire thing, just one little piece. So what we're going to take that one gene, one segment of it, and based on that, we're going to build a brand new little chunk of RNA. So that process, again, let me extend that a little bit more, of taking only one template, only one of the strands, and only one small portion of it that allows us to build one little piece of RNA. So that whole process is what we call transcription. Let's put it over here. Whoops, I added an arrow by accident. Now, there's a couple of little bit of notes to talk about in here. Is that, well, how do we know where to start to look for that gene, and how do we know when to stop? So how do we know that that's the beginning of that guy and that's the end? And so for that, we actually have positions. There are specific sequences that allow us to do that. So for example, the beginning point is called a promoter sequence. And then the ending point, so we know where to stop, is called the terminator sequence. So there are regions, let's see if we can put these a little bit better, to tell us exactly where these things go. So to make it a little bit simpler for me, there's a little piece at the beginning that tells us where do we select the right gene. And there's a little piece at the end that tells us also where to do that. So the beginning of it is called the promoter sequence. The end of it is called the terminator sequence. So whenever we want to decode one of those genes, we know where to start and we know where to end. Okay, and so there is a protein that does this whole job. This process of transcription is done through a protein also. So via a protein, actually I'm going to type it up, but this is called an RNA polymerase. So let me kind of add it over here. So it's through a protein, an enzyme specifically that does this, called RNA polymerase. And so this is kind of the overall process of converting the library or collection of these guys, this whole collection of thousands of these guys, and only selecting just one of them so we can just build one protein, one functional unit, right? And so again, to search through the thousands and thousands that exist, we need to have a place where to start and where to end. And so to do that, we have very specific timings and plannings on how to actually do that through these cool little enzymes called RNA polymerases that know when to start and when to end. Now, this is also something that there are a few little exceptions here and there in which, for example, once you're done transcribing your RNA, there are some modifications you can do. So let me kind of add it to here. Is that once transcription is over, so I'm just going to write it as TCR, or sometimes we write it as TXN, depending on who abbreviates it. You usually need to modify the RNA. In other words, you kind of need to alter it just so it can do its final functions. And so this actually changes dramatically between organisms. And so this is known usually as post-transcriptional modification. In other words, once you're done completing and making your cool little tiny chunk of RNA, you still need to tweak it just a little bit more. You need to edit it, basically. And so the editing actually takes place differently between organisms. In eukaryotic organisms, there's three main steps. In most prokaryotes, times it happens just very kind of loosely. And so I'm basically going to highlight one. Those are the main ones you probably want to know for right now, although there's definitely usually more. And so for the eukaryotes, humans, dogs, chickens, all those guys, once the little chunk of RNA is made, the post-transcriptional modification involves three things. One of them is called capping, adding a tail, called a poly-A tail, and the final one is splicing. Whereas in prokaryotes, none of this really happens, although there are some small modifications of them. They're called usually methylations, in which some of the bases, some of those A's, G's, T's, and C's get a little tweaked. Now, to kind of explain that, though, then we're going to have a little bit of a side discussion over here. Let me see if I can get another slide going on over here. We're going to call this eukaryotic post-transcriptional modification. So let's give you the slide, if this thing listens. Nope, it's not listening to me. Hang in there. There we go. In which we're actually going to take that little chunk of RNA that we just made. So let me take that. I'll bring it up over here too. Here's our chunk of RNA that you guys remember from the previous slide. And what I'm going to do is actually, I'm going to expand it and make it bigger. Whoops, too big. Sorry. All right, let's keep it there. Seems to work. Into just a big version of that guy. And so to this little chunk of RNA, we're going to tweak it. We're going to modify it. So the first thing that's going to happen is right at the beginning of it, we're going to add a little hat. So right here in there, we're actually going to add an extra little base kind of backwards. So this is called a cap. And so that little cap that you place at the beginning of it kind of protects it from being degraded and does this one additional function that I'll explain in half a second. Now there's a second thing that happens. At the end of this guy, trying to modify this. At the end of this, right at this little piece, we actually add a tail. And so that tail, pretty much the same thing, it kind of protects it from being broken down. And so both the little cap that goes at the beginning and the tail that goes at the end are protective mechanisms to prevent the breaking down of that RNA, that precious interpreter that is going to help you build your final product. Now, the third step that occurs is the last one that both of these guys contribute. It's called splicing. And so for splicing to actually take place, you actually require both of these first two. Okay, so you actually need the presence of a cap and a presence of a tail in order for you to actually perform splicing. Now, what is this so-called splicing, which is what most people kind of have heard, and especially in sci-fi movies and things like that, but it doesn't quite mean what people think it means, is that splicing is the removal of extra sequences of RNA that we don't need. Okay? So what's going to happen is we're going to take that little chunk of RNA. So I'm going to put it over here. There it goes. I'm going to make it a little bit bigger just for the sake of it. And what's going to happen is that little chunk of of RNA is actually divided into little pieces. So let me remove that. That way it doesn't get confusing. There's pieces in there that are useful, and there's pieces that we don't use. So let me kind of copy them over here. So some of those regions in there, for example, let's take these guys over here, are not needed. So let me kind of shrink this over here so I can make a little more sense. It's not letting me do that. Hang in there. There it goes. Are not needed. So let me delete this guy. And then there's definitely a region of them that are definitely needed. So let's remove that part. And so what ends up happening during the process of splicing is that we only keep the needed parts and we get rid of the unneeded ones. So we can take these two guys, and then what we do is piece them back together. Let me create a little arrow between these two. There we go. And so we kind of connect these guys together to build one new piece. So normally what happens is our little chunk of RNA ends up being a little shorter. A little bit of a shorter piece. And so the process of you pretty much editing out the unneeded versus adding the needed pieces in there is called splicing. And so the ones that we need have a fancy name. They're called exons. And the ones that we don't need are called introns. So more or less what's happening is that we modify the RNA, that one little piece of gene that we just made into RNA, and then we tweak it a little bit. We put a little cap, we put a little tail onto it, and then once we put those two on, we edit by cutting out the things that we don't need. So we excise or cut introns out, and then we take the ones that we do need, the exons, and then splice them back together. And so ultimately, what this leads is to a new term of RNA. I'm looking to insert this little title. Is usually referred to as a mature messenger RNA. Now, we don't usually refer even to the term mature most often. Now we just end up calling it RNA, and that's about it. But again, this is very unique to eukaryotes. This does not take place in bacteria, for example. This is very unique to all things that have a nucleus, basically. So that was a little side story from what we were dealing with originally during transcription. Getting this guy to move. There we go. So we just kind of finished discussing in the previous slide that we take our DNA and duplicate it or replicate it into two semi-conservative pieces of single strands of DNA. And then we introduce the process of transcription, in which we only take one of those strands as a template and take one section of that entire strand called a gene and then produce a tiny little piece of RNA from it. Now that RNA needs to be modified or tweaked basically or edited, which is what we just kind of finished covering here for most eukaryotes. So again, this happens in animals, this happens in fungi, this happens in protists, but again, it does not take place in bacteria. Now, that being said, we can enter our final step of once we've interpreted it, the message, now we can take that information and convert it, not necessarily convert it, let me correct that, and build a protein from it. In other words, we're going to take the mRNA and turn it into protein. A process that you guys know as translation. So as we continue to cover the final step in the Central Dogma, this idea that we've now taken the original instructions, so the DNA, and replicated it to make more of it, and then we also decoded it so that we can understand it a little bit better. We are now going to take that message, so I'm going to write this in red because I try and color code it a little bit more, which we know as the RNA, which possibly has been edited through capping, tailing, and splicing, and now we're going to convert it into some protein. That's the idea, through this wonderful little process known as translation. So let's take what we actually had from the previous slide, just to kind of help us with what we did. I'm going to take the exact same image that we had before. Bring that guy in. All right, there he is. And so we're going to take that little segment or piece of RNA that hopefully has been edited. So I'm going to make a little copy over there. And so I'm going to expand it really quick so we can read it a little bit better. And then what we're going to take is a starting point and an ending point on that piece of RNA. So let me take the same stuff we did from the previous slides. I'm going to try and copy and paste it. So there they are. So the same stuff we did before, right? So I'm going to take a sequence called a start codon. So I know when to start making this. And I'm going to get another sequence. Trying to make this work a little bit better. Called a stop codon. So I know when to stop. As silly as that sounds. And so from these sequences, that translation process occurs through a really cool giant enzyme called a ribosome. Now, for those of you who remember that from topic C, is your ribosomes were those one set of organelles from either just in the cytosol or in the rough ER that were in charge of building protein. This is a little bit of an extra bit of review from that point. So for this process, we need to know where to start, where to end, so that our cool little ribosome guy comes in and does that. Now I'm going to attempt to draw a little ribosome by creating two little circles over here. I'll put one, and then I'm going to put a second one. Okay, and I'm going to shape them. So remember that your ribosomes are composed of two pieces, one large subunit and one small subunit. And so they often end up being drawn like this. And what you kind of have one big little oval and one smaller one. And so what will happen is that ribosome, I'm going to put that guy over here so that way you can identify them. That guy. Will actually land at that start codon, right? And then it'll continue to read that entire process from the start place all the way down to the stop place. That's what it's doing. So it'll kind of proceed along. I'm going to make this a little bit smaller. Step by step until it makes it till the end. and then it ultimately kind of goes away. Now, as it's reading that small segment of the RNA, it starts putting together amino acids. So for that, I'm actually going to create another little piece and put these in green in a moment. Make this small. Little pieces of amino acids, and I'm going to rename this guy so you can see it. They will ultimately all come in, and based on what your ribosome is reading on that RNA, right there, right, it'll start assembling them. And so I'm going to make this guy really, really tiny. And so what it will do is start assembling them and then putting them together. Little by little, pieces of those amino acids kind of being assembled until ultimately you have this very, very long chain of these guys. I'm going to draw it over here on the lower left-hand side. You get such a long chain of these guys, and it's just probably easier to illustrate by a green arrow now. You get such a long chain of amino acids that ultimately then we enter that final step that we started with the lecture. Hopefully for most of you guys remember this, is that enters this thing called protein folding. It looks like I lost my little writing over here. Hang on. There it goes. And so for those of you who remember this part now, that turned into that wonderful portion that we call primary, secondary, tertiary, and quaternary structures. So let me kind of get that out of the way. Primary, secondary, tertiary, and quaternary structures. So right at the beginning of this topic, we had told you, let me get my symbol for degree over here. That way I can always add it. I have to do a little quick search to find that guy. There it is. Primary, secondary, tertiary, and quaternary structures. And so for those of you who remember that, once we actually end up building our protein out of those amino acids, then we have our primary sequence, which then gets folded into secondary structures. Those were your alpha helices and your beta sheets. And then ultimately those secondary structures get built into your tertiary structures, right? Which give it the overall shape. And then sometimes they get assembled into more complex structures between multiple sequences. Those were your quaternary structures. So this was what gave you the ultimate three-dimensional shape. And I assume that most of you remember this just because we finished talking about this in the last lecture, is that ultimate three-dimensional shape is what finally gives us our function. And just like we just saw in the growth lecture, right, that function can then be altered by things like pH and temperature and so on and so forth. So we can go back really quick to this very, very beginning, one of the slides that I showed you over here. So let's see if I can get it. This guy right here. And so pretty much we've gone through this entire process. We've taken the three main components, DNA, RNA, and protein. We've covered their overall processes, replication, transcription, and translation, as a way to demonstrate that the material that we're all built out of is stored into these pockets of, or little books of instructions called genes that are all collected into this giant library called DNA. That as we read individual books, they need to be interpreted into these decoded messages called RNA. And then ultimately, once we understand how to read those books, we can actually build things out of them. And this is what ultimately gives us proteins. And it is those proteins that ultimately give us functions that do stuff for us. So these are the things that keep us alive. So this process from DNA into RNA into protein is what we call the central dogma. Okay, so now that we have the overall concept of making protein, this whole process from DNA to protein, known as the central dogma, also gets a little fancy name. We call this protein expression, this idea of making protein, right? And so most of the time, most of the genes are expressed, I should say. So the proteins are produced or made most of the time. They're actively being made. And so we call that a constitutive process. So proteins that you always need, that they're always being employed, are referred to as constitutive proteins. However, there are some that you don't always use. So you only need them at certain times or certain conditions. Think about, for example, every single time you need to digest your food, for example. You're not always digesting. You're not always trying to extract energy from things. So you only make certain proteins when you consume food. And so those are referred to as facultative proteins. Proteins that you only make or you only build when you need them. Now, that'll take us to a very interesting process, because in most eukaryotes, that's pretty standard. But in prokaryotes, they've kind of developed this very unique way of making proteins a little bit more efficiently. And this is what we call operons. Now, this only occurs in prokaryotes, so this only happens in bacteria. Humans don't do it, chickens don't do it, lizards don't do it, fungi or plants don't do it, just bacteria. So your good old E. coli and those guys, those are the ones that do it. Now, what these organisms do is, rather than making one protein at a time, which happens to be a fairly long drawn out process as we just finished covering, bacteria can just do it a little bit faster. They basically do is put a bunch of genes together at the same time, connected one side by side, as a way to save time and energy. Basically, killing more than two birds with one stone. And so what they're doing is creating something called a cassette, or better known as an operon, in which you bring multiple genes, and they're all basically expressed, translated, if you will, at the exact same time. The idea is, again, saving time, saving energy. Now, the interesting thing about this is that there are requirements. These are not just kind of built willy-nilly. There's rules involved on how to make this happen. And so we introduce a brand new term that you haven't seen called an operator. The term operator in this case is a very specific protein involved in literally operating or controlling how these genes are made. Like I said, most of the time they're constitutive, and some of them are facultative, meaning most of the time they're being made, and then there's a few that are only made as needed. But bacteria got a little bit smarter. They decided that they control multiples of these guys at the same time by this process called an operon. And we talk about two classic operons that everybody gets to learn about in at least microbiology. And the way they work is by being controlled again at the same time. So again, there's always rules involved. So let me kind of introduce really quick what these are based on the examples. There are operons that basically are always off, meaning they're not doing anything, and must be turned on when needed. And there are other operons that are always turned on, meaning they're always making proteins, and then they must be turned off again when needed. So the always off and then get turned on are called, is the example that we have here is called the lac operon. This is also known as an inducible operon. So let me add it here. Let me add my little parentheses over here. So that's one version of it. So that means it can be turned on. And then the ones that are always on and must be shut down, basically, are called repressible. Let me put that back in there. And that's known as the tryptophan operon, also known as the trip operon. So what does these entail? Let's see if I can get it here. Right? The lac operon on the left-hand side, we're seeing here, basically is always turned off and is only turned on under a certain set of conditions. The conditions are as follows. The lac operon is designed to produce enzymes to process lactose, hence the term lac operon. But there's no reason to have it on because most of our sources of energy come from glucose. So basically the lac operon is always off. We don't really need it most of the time, or at least bacteria don't really need it most of the time. However, every now and then you do run out of glucose, and you need to find an alternate source for carbohydrates, for sugars, for energy. And so if there is lactose present, an alternative towards glucose, then you can actually turn on the lac operon. And so there's two conditions that are required for the lac operon to be turned on. One, there needs to be glucose gone. Basically, there needs to be an absence of glucose. And there definitely needs to be a presence of lactose, the other source of energy. Those are the two key conditions. Now, the idea behind this is actually quite simple. You don't want this guy to be on because you don't want to be wasting energy and materials for something you don't need. As long as there's sugar around, as long as there's good old glucose around, this guy is not needed. However, if you run out of this guy, still, there's no reason to turn this guy on unless there's the presence of the alternate sugar, lactose. So for this to be turned on, in other words, for this to be induced, you require the absence of glucose and the presence of lactose. Now, its converse operon is known as the trip operon or tryptophan operon, the one that you're seeing on the right. Now, in this case, the trip operon is always on, meaning that it's always producing tryptophan. It's always building the amino acid tryptophan. Now, why would you need to turn it off? Well, the only reason why you turn off this guy is when you don't need tryptophan. So most of the time, it's always on and must be turned off, again, under certain conditions. And in this case, the condition is if you have excess tryptophan present. So you turn it off only if you have extra amino acids there present. Otherwise, you'll always have it on. The second you have excess tryptophan, this whole guy gets repressed or turned off. Now, what you're actually seeing here on the little doodle is just an explanation of exactly what we saw earlier. So I can actually copy this guy and put it off to the side so you guys can see this. So let's take a couple of genes. Actually, let's take a little piece of DNA more than anything else and use that as our template. So let me take our basic one. I'm going to take this guy. And so for those of you who remember, I showed you something like this, right? So we had a little piece of DNA and we had one gene to make one piece of RNA. Operons, on the other hand, don't do that. Instead, they actually take multiples of these guys side by side. This is what we call an operon, a collection of multiple genes, all one back to back to back behind each other. The idea behind this is that rather than producing them individually, you can kill 3 birds in 1 stone, if you will, sometimes up to 5 birds in 1 stone. And so what will happen is that because this is a fairly large system, you usually have something preventing it from working. And so I'm going to introduce something called an operator. Whoops, I used the wrong color. I was trying to use purple here. There we go. There's usually something at the beginning of this, either preventing it from working or turning it on or turning it off. This is what we call an operator. All right, so I'm going to bring this guy next to this guy over here. That way you can kind of identify that guy right there. Now, what normally happens is, again, you don't really want to waste too much energy here. So you only turn this on or off as you need it. For the case of the lac operon, the 3 genes involved in producing lactose are always off. We don't need to waste energy making this. So this is a lot of material. There's a lot of energy to build this. So there's this little protein right here called an operator blocking it. It's basically turning it off. Now, in the event that you run out of sugar, you run out of glucose, and you also have the presence of lactose, this little operator can easily just kind of fall off, and then you can now produce all 3 of those proteins necessary to process lactose. But again, you require both conditions. Glucose has to be absent, and lactose has to be present. That's how you make all of those particular genes. Now, the alternative for the trip operon is slightly backwards, or in reverse. All of these guys are always turned on and always making tryptophan. But when you have excess of it, then you don't need these guys to be working. So if there's excess tryptophan, there's excess amino acids, then those guys get combined with the operator, and then they get in front of the actual operon and then prevent it from working anymore, and therefore you decrease the production of tryptophan. So on one side, you turn it on, and the other side, you turn it off. The whole premise behind this is that bacteria use multiple genes, multiple proteins at the same time to save some time and energy. But these efficient ways of doing that require some extra little steps of control, basically something called an operator that prevents it from moving or going on or turns it on and turns it off as needed.